Bismillahirrahmanirrahim. My name is Saima. I am student of M.phil first semester. My registration number is MB32023103. Subject Molecular Biology. Presented to Dr. Niaz Muhammad. My topic of presentation is fermenter. Outlets is fermenter, bioreactor, size of fermenter, construction of fermenters, types of fermenters. Now, what is fermenter? It is enclosed container with adequate arrangement of aeration, agitation, temperature and pH control and drain or overflow vent to remove the waste biomass of cultured microorganism along with their products. It is a device in which a substrate of low value is utilized by living cells or enzymes to generate a product of higher value. It is extensively used for food processing, fermentation, waste treatments, etc. Bioreactor. All bioreactors deals with heterogeneous system dealing with two or more phases, that is liquid, gas or solid. Therefore, optimal condition for fermentation necessary efficient transfer of mass, heat and momentum from one phase to another. Generally, 20 to 25% of fermenter volume is left unfilled with medium is head space to allow for splashing, foaming and aeration. The fermenter designs varies greatly depending upon the size and fermentation for which it is used. Bioreactor. A bioreactor should provide the following: agitation, aeration, regulation of factors like temperature, pH, pressure, aeration, nutrients feeding, liquid level, etc., sterilization and maintenance of sterility, and withdrawal of cells medium. Modern fermenters are usually integrated with computers for efficient process monitoring, data equation, etc. Size of fermenters. The size of fermenter ranges from 1 to 2 liter laboratory fermenters to 5 million liters or occasionally even more. Fermenters of up to 1.2 million liters have been used. The size of fermenter used depends upon the process and how it is operated. Construction of fermenters. This is the structure of fermenter shown in the figure. It is a steel stainless fermenter. Large scale industrial fermenters are almost always constructed to stainless steel. A fermenter is a large cylinder closed at the top and the bottom, and various pipes and valves are fitted into it. All of the process and structures are shown in the figures. Since most industrial fermentation process is aerobic, the construction of a typical aerobic fermenter is the following. It have a cooling jackets. The fermenter is fitted externally with a cooling jacket through which steam or cooling water is run. Cooling jacket is necessary because sterilization of the nutrient medium and removal of the heat generated are obligatory for successful completion of the fermentation in the bioreactor. This is the structure of fermenter. Aeration system, critical part of the fermenter. It is in a fermenter with a high microbial population density, there is tremendous oxygen demand by culture, but oxygen is being poorly soluble in water, hardly transfer rapidly throughout the growth of medium. Two separate aeration devices are used to ensure proper aeration in fermenter: spargers, impellers. The stirring accomplish two things: it makes the gas bubbles, it makes the microbial cells through the liquid. Baffles. The baffles are a metal strips normally incorporated into fermenters of all sizes to prevent the vortex and improve aeration efficiency. controlling devices for environmental factors. Environmental factors that are frequently controlled includes temperature, oxygen concentration, pH, cell mass, level of key nutrients and product concentration. Uses of computer in a fermenter. The main use of computer in a fermenter is monitoring data equation, data storage and error detection. Now types of fermenters. Following are the five types of fermenters: air-filled fermenter, continuous tank bioreactor, photobioreactor, bubble Coleman fermenters, fluid, fluidi, fluidi, fluidized based bioreactor, packed based bed bioreactor. Thank you.